researchers from Illinois State University in the United States were interested in whether overexpression of particular motor transport proteins will affect the length of cilia in cells. So to investigate this, they transfected cells with their motor protein and then fixed and stained them with DAPI, two markers which will help them uniquely identify the presence of cilia, as well as a marker that lets them visualize the level of expression of their motor protein. The task here was to measure the motor protein intensity in a perinuclear region of the cell, and if that cell has a cilium associated with it, measure the length of the cilium. In addition, of course, cilia associated with non-expressing cells also had to be measured. So for this analysis, we used CellPose to segment both the nuclei of all of the cells and the cell bodies of the cells that were overexpressing the motor protein. We used random forest pixel classification using LabKit in Fiji to detect the cilia, again using the two markers that together identify cilia uniquely. From there, we needed to associate each cilium with the correct nucleus that belongs to the cell that houses the cilium. And this was particularly challenging because this may not always be the closest nucleus in the case of, for example, very elongated cells. Once we'd done that association, we needed to measure the corresponding cilium length and the motor protein expression in a perinuclear region. The most difficult part of this project was actually just the coming together of lots of different pieces. So for example, I mentioned earlier that we needed to uniquely associate each cilia with one nucleus, and this may not necessarily be the closest nucleus in the case of very elongated cells. And so we had to think about a way of associating nuclei with cilia uniquely. Another issue was in densely packed cells, the perinuclear regions might overlap, or in fact, the perinuclear region of one cell might intersect with the nucleus of another cell. The first was, as a bioimage analyst, it's always very nice to see a script that you've been working on with many, many moving parts come together and actually deliver the results that you thought it would. And the second is delivering this script to the users and hoping that this can really enable them to progress in their research and discover new biological insights. If you have a particular bioimage analysis problem that's really blocking progression of your research, this is your opportunity to collaborate with experts in artificial intelligence and bioimage analysis who can help you overcome this block and progress in your research.